All right, so we have completed the cell cycle in our previous classes, and we have talked about interphase, we have talked about mitotic cell cycle, then we have talked about cytokinases. So apart from that, now what we have is a different subtopic, which is of telomeres. So we are already aware of this structure by now that there is this centromere, which is the constricted site that holds these two sister chromatids, and you do not need to worry about the long arm or the short arm. Okay, this is not of your concern at this point, but the new thing that we are going to study is telomeres, which you can see it is something which is towards the end of your um towards the end of your chromosome. Okay. So we are going to study about this. Okay, what exactly this is and what is the function of telomeres. So initially what you're supposed to write is its location, that it's present at the tip of DNA or it is present at the tip of your chromosome, like you can see. Okay, so this is very self-explanatory that the location of telomeres is towards the end or towards the tip of your DNA or chromosome. Then your telomere elongates the DNA. So one of the functions of telomere is to elongate or to lengthen up your DNA. And how telomeres are built, they're built with the enzyme action of telomerase. Okay. So telomerase is responsible to build telomeres. And what exactly telomeres is? It's something which is present at the end of DNA. Then, at this point, I know you are unable to understand what DNA polymerase is because we have not studied the chapter nucleic acid so far. It's the next chapter that we'll study. So we'll talk about DNA polymerase in detail. But for now, you can just understand that it's an enzyme. DNA polymerase is an enzyme which copies the DNA, but replicated one is a short one. You'll understand the proper working of DNA polymerase in the nucleic acid chapter. But for now, just try to keep this in mind that whenever your DNA is replicated, meaning we have already studied about the process of mitosis. So I'm just saying that when this DNA molecule or when this DNA structure replicates itself with the help of DNA polymerase, it is shorter in size. I also mentioned this in previous class that the new daughter cell which is made, it's smaller in size. So anything which is newly synthesized, it is going to be shorter or it is going to be smaller in size. Then what exactly happens is that when your DNA is replicated and it's a short one, then your replicated DNA will result in loss of genetic information. So as you can see that there's so much information stored in all these arms, right? But like I'm saying, when this will replicate itself, it will be shorter in size. So when it will be shorter in size, it means that it has lost few of the information, few of the genetic information which was held in these arms, right? So this is exactly why we say aging is related to telomerase working. Like you see, if you have grandparents in your home or if you have parents in your home, then you can feel that they tend to forget the things. This is because their DNA, whenever their DNA will replicate, it will be shorter in size. So with as your age progresses, the working of telomerase results in loss of genetic information. Hence, people tend to forget things because their DNA gets shorter and shorter over time. So, telomeres are responsible to elongate this DNA, but they do not have any, what do you say? They do not contain any genetic information. They're just there to elongate it. Okay, so with the presence of telomeres or with the presence of telomerase, which is the enzyme that produces telomeres. So we say that aging is related to telomerase working. So as your DNA will get shorter, your aging will progress and you'll tend to forget things. ठीक है? Clear है so far? Sometimes they also ask you, okay, draw a DNA with complete components. Okay, so now that you're aware of telomeres, so you'll label telomeres as well in your chromosome. Okay, this may come for 4 marks. So you are expected to draw a centromere, you are expected to draw a sister chromatid, you're expected to draw telomeres at all four ends. ठीक है? Do not do this thing कि एक जगह label कर दिया and that is it. No, you have to draw on all four sides. And we'll study about more components of the chromosome. Then you'll have to draw all of them if you are asked to do so. ठीक है? जी उस्मान, do you have any question?
यस। मेसी जो टेलोमरेज है ये एंजाइम है।
एंजाइम है जी। हर चीज जिसके आखिर में ASE आता है, इट्स एन एंजाइम।
ठीक है।
Now, so now the other thing that we have to study, so these are basically the components that you need to keep in mind while you're drawing the chromosome. So the other component that you're expected to draw is kinetochore. Okay, learn this word, kinetochore. So kinetochore is the attachment site on centromere for spindle fibers. So we talked about these spindle fibers that they are produced during cell division and they work like a rope. They pull the chromosomes towards the opposite poles. So kinetochores, you can assume kinetochores is the hanger, uh, like we hang our clothes in hangers, right? So you can assume kinetochore to be the same hanger and it's present on your centromere. Okay, you cannot see it properly. We just draw it like this, like this, because beach में तो you'll find centromere. Up on sideways, you need to draw kinetochores like this. Okay. So kinetochores, and then you have to label them. That kinetochores are the attachment site, and they provide attachment to spindle fibers. And we have talked about the function of spindle fibers, that they pull the sister chromatid to opposite poles during anaphase. This is it. For kinetochore, just remember the fact when you draw your chromosome, you'll show that this is your centromere. ठीक है, on sideways, you just need to label that these are your kinetochores. ठीक है, this is it. You are not expected to write about kinetochores or about the function of kinetochores. You are just expected to draw a diagram of kinetochore. ठीक है, make sense?
यस।
Then you have non-kinetochore. So you're not expected to draw these, but you are expected to write the functions of non-kinetochore. Okay. So these are the four functions which I've extracted from your marking scheme. So just try to learn as it is because these are your keywords eventually for non-kinetochores, and they highly appreciate these words. So these are the words that are accepted in your marking scheme. Okay, so non-kinetochore are the fibers that run from pole to pole and play a role in cell elongation. They align the chromosome at metaphase plate, they move the chromatid, and they guide movement of chromosome during mitosis or meiosis. Sometimes when they ask you this question that write the functions of non-kinetochore for 2 marks, then take this point as your preference point, meaning that write this one as your top priority. Okay? That non-kinetochores are responsible to move the chromosomes during mitosis or meiosis. And the other, you can write any other point, whatever is convenient for you or whatever you can remember. But if they're asking you for 4 marks, then you need to write all of them. But if they're asking you for 2 marks, then this should be your preference that you need to write here. It is guiding the movement of chromosomes during mitosis or meiosis. So basically, these are the fibers that run from pole to pole. So we have studied the cell has poles, right? And these are those poles which guide the movement of chromosomes during anaphase. We talked about the fact that your chromosomes become V-shaped, right? So they become V-shaped during anaphase and then they're pulled towards the poles. So these non-kinetochores are somewhere present over here, and these are fiber-like structures that are present from pole to pole. And since they're present at pole sides, this is why it is helping your chromosomes to move during mitosis and meiosis. And then it's also responsible to align the chromosome at metaphase plate. So it's a similar function that it's guiding the chromosome where it has to place itself, and then it moves the chromatid. So since it's present here, spindle fibers are coming along the poles. So all of this contributes to the movement of chromosome. Then you're sometimes asked to write the importance of mitosis for 4 marks. So these are again your keywords that should be in your script, that it promotes genetic stability, that your daughter cells are identical to parent cells as they are clone of each other and have same genetic makeup. So now you have seen how mitotic cell cycle takes place. So you are well aware of the fact that how the genetic makeup during the process of mitosis remains same. It does not change because cell has replicated itself from the same chromosome. Okay. Then damaged cells are replaced by exact copies. Since we are talking about mitosis, then this is well justified that damaged cells are always replaced by exact copies. Then asexual reproduction takes place in plants, and sometimes your mitosis can help to regenerate parts. So it's written for tissue, but through mitosis, your liver can regenerate. So liver is the organ which can regenerate itself in 4 weeks. Okay, so regeneration of parts is also important. All right. So the next concept is of stem cells. So stem cells are basically your cells, and then these stem cells, they undergo the process of differentiation. Differentiation means that after the process of differentiation, they acquire certain and certain functions. Okay. So stem cells undergo through the process of differentiation, and then they have three different types of differentiated cells. Number one, totipotent. Number two, pluripotent. And number three, multipotent. So as far as totipotent stem cells are concerned, it's just a combination of a sperm and egg which results in a zygote. So zygote is the example of totipotent stem cells. That when sperm and egg, they combine to form zygote, they form a totipotent stem cell. Then eventually when your zygote grows, it becomes a embryo. Okay, so when it becomes embryo, or when different sort of tissues of embryo combine, because initially the whole process has to take place for the proper formation of what do we say, for the proper formation of a fetus. So initially it has a cell level, right? These are two cells. Then we have tissue, and then eventually we get the proper organ system. So tissues of embryo contribute to the formation of pluripotent stem cells. And at this point, that all of us are adult. Now we have multipotent stem cells. So multipotent stem cells means that from zygote to embryo to fetus, now we have all the cells that are differentiated, and they have their certain and certain traits. They are following their certain functions. For example, your bone marrow is responsible to produce RBCs, then your heart is working properly, so you have cardiac cells. For brain, you have proper neurons. Then for skin, you have dermal cells. Then similar is a process that your bone marrow will keep making RBCs, right? Because RBC has just a lifespan of 120 days. So there are those stem cells, there are those multipotent stem cells present inside us in our bone marrows that are responsible to produce RBCs, which is a lifelong process. Okay. So stem cells, what are they? They undergo the process of differentiation, and then we have three different categories of differentiated cells: totipotent, pluripotent, and then multipotent. Initially, we have totipotent stem cells, which means that with the combination of sperm and egg, zygote is formed. Then eventually when zygote grows, it becomes an embryo. So then we say that it's a pluripotent stem cell. And now during the adult stage of life, or during like right after a baby is born, then we can say that it has multipotent stem cells because all of the cells are working in a proper condition with their associated functions. Like heart is not going to transmit the impulses, right? Heart is just responsible to pump the blood. Similarly, brain is going to transmit the impulses, or it's going to process the impulses. And then bone marrow has its certain function that it will make RBCs. ठीक है, उस्मान क्लियर?
जी मिस।
ठीक है. So now the last subtopic for this chapter, it's of cancers, and they do not ask much about cancers, but just a superficial idea would be sufficient for you at this stage. That why we are studying cancers is because this is a chapter of mitosis, and when uncontrolled mitotic cell cycle happens, meaning that your cell cycle is not regulated, it keeps on happening. Your new cells are being produced again and again without being differentiated. So we say that your mitotic cell cycle has become uncontrolled, resulting in cancer. So basically, initially your cell has to decide whether should I divide or should I not divide? Is a new cell required or is a new cell not required? Only then mitosis happens. Mitosis is not happening every hour of the day. It just happens when it's needed. ठीक है, the normal human being. But in a cancer condition, your mitotic cell cycle becomes uncontrolled. That your cell is unable to identify that whether whether a new cell is needed or it's not needed. So uncontrolled mitotic cell cycle keeps on happening, which results in abnormal cells, and these are the cells which do not die. Okay. So these are the cells which do not die. Like in a normal human being, when cell is ruptured or something happens to a cell, it dies. Okay. So that process is known as apoptosis. If you want to write it, you can write it. Apoptosis is the process in which the cell dies after completing certain age. Okay. So what did I say? I forgot the word. Apoptosis. Process. Because apoptosis is the process where your cell dies under the condition of rupture or under any condition when it has fulfilled its lifetime. Okay. So in cancer condition, the abnormal cells are being produced, and the irony is that they do not die. So apoptosis uh, uh, completely pause completely pauses itself during cancer. Okay. So imagine that your cells are so many cells are being produced in abnormal mitotic cell cycle that those cells do not even get the chance to differentiate themselves. And what did I say about differentiation right now? That differentiation is a process in which your cell gets a certain function. Okay, I am a cardiac cell, now I have to pump the blood, or I am a neuron, I have to transmit the impulses. Although both of these cells are not regenerative, if your cardiac cells dies, then no other cardiac cell can be produced. And this is exactly why sometimes heart conditions are chronic in nature. And similar is the case with your neurons, that they are not regenerative. That and eventually, if your neurons they are dying off, then you get different sort of diseases like what's that disease known as Parkinson's disease. If you've heard about it, it's about neuronal degenerative condition where neurons die, and eventually people start to forget things. Okay, so the example of cardiac and neuronal cells is not appropriate. This is exactly why I give the example of skin cell, because it's easier for us to relate. So so cells are being produced in such large quantity that they do not even get differentiated, and those cells are abnormal, and then they do not die. Okay. And why exactly? Sometimes they ask you how cancers occur. So you can write that it's a mutation oriented process where carcinogenic agents like X-rays or tar or color food or gamma rays or excessive UV radiation, these things can cause mutation, and they can result in your mitotic cell cycle being uncontrolled. Yeah. So the most important for you to understand is colored foods. Right after studying this chapter, and when I came to know that all the colored foods are cancerous in nature, or they have a carcinogenic nature, I have, Alhamdulillah, now I'm not using any of the colored foods in my life. I was a big fan of donuts, but all those sprinkles and those color. they're very health they have a strong health hazard, so try to avoid them because they are mutagenic agents and they can cause mutation in your cell cycle. And then you need to remember that your cancer lumps are stony hard and sometimes they're painless. So if you think that there is a cancer, then you can see okay, if you have any lump, and lump is a mass of cells, and if you think that's if you press it and you feel that it's stony, then there's a chance that a cancer is being formed. And those lumps are, there's sometimes they're painless, sometimes they can be painful. Then there are two types of cancer. So there's a primary cancer and then there is a secondary cancer. So primary cancer is the original site of cancer. For example, if someone has a cancer of liver, so eventually, initially he had this cancer of liver. So liver is the primary site, meaning that it was the original site of cancer. But cancers do have this tendency to spread. So if the cancer has spreaded and it has moved from liver to the entire body, then we'll say that the cancer is now secondary. So secondary means that it has spread throughout the blood system or lymphatic system. And we have two types of cancer. So these are the two types of cancer which describe this location. Okay. Then these are the words which have the bigger umbrella. We have benign cancer and we have metastasis wala cancer. So benign cancers do not spread. Okay, and this sort of cancer is usually in the brain. That it's a benign cancer, it does not spread through your blood system or through your lymphatic system. But your metastasis cancer can spread and it results in a secondary cancer. Meaning if there is a cancer in liver, then eventually it can spread in the entire body. And sometimes they ask you to write the treatment of cancer for 3 marks. Then you can write these pointers that a surgery may be performed after properly examining the condition, then radiotherapy on top priority, then chemotherapy. So